My name is Naveen. I'm a graduate student at Virginia Tech. Today I'm going to deliver my talk on phenotyping and genotyping RIL population of Virginia type peanut for drought tolerance in Virginia, Carolina. Here is the outline of presentation. Firstly, I'm going to talk about the justification, why there is a need to study drought in Virginia, Carolina region, followed by hypotheses and objectives framed in our study, then material methods, results, and future plans. Here are a few factors that drive us to study drought in this region. To grow and yield profitably, peanut require a regular average weekly supply of water ranging from 30 to 50 millimeters or mm. This graph shows monthly variation of daily mean temperature and weekly mean precipitation over VC region in last 30 years. So adequate rainfall early in the season stimulates vegetative growth, but the critical growth stage of water requirement in peanuts start from beginning of bagging to pod filling in the month of July and August. And at this time, peanut require about 75 mm weekly rainfall. But the rainfall is generally about 25 to 30 mm per week in this region, leading to drought stress. Drought is also supplemented by high temperature in July and August when pod and seed growth are intense. The second factor is sandy soil in this region. Peanut prefer to grow in sandy soil as sandy soil is favorable for pegging and pod formation. However, at the same time, sandy soil has lower water holding capacity, which make peanut more prone to drought stress. Under drought conditions, irrigation can be supplemented, but 85% of the peanut acreage in VC region is under rainfed condition, and only 15% of the peanut farmers are irrigated. So a report from Dr. Bolota group stated that drought in VC region cause annual losses up to $50 million to the peanut grower. So under all these conditions, the most reliable and economic solution is to develop drought tolerant cultivars. To achieve this goal, we hypothesize that sufficient genetic variation exists in our germplasm for physiological traits that contribute to drought. But to precisely identify and validate the contribution of those traits, we frame our objectives as phenotyping and genotyping of recombinant inbred lines for drought tolerance. Moving towards the material and methods. For phenotyping and genotyping, we use recombinant inbred population that was developed by crossing large-seeded, high-yielding Virginia type parent N0808 with the Spanish type drought-tolerant parent PI 58505. And the trials were conducted at two different locations, one at Lewiston in North Carolina and second at Suffolk in Virginia. So phenotyping is done by taking measurements on Ndvi, Ctd, wilting, and SPAD in the field at different growth stages and drought severity. Ndvi is a vegetative index that relates to chlorophyll content based upon light absorption. It tells about the overall health of plant and used effectively in predicting the biomass and yield under stress. More the Ndvi value, more is the plant health as shown in the picture on left side. We measure Ndvi with handheld green seeker. Similarly, canopy temperature depression was widely used as selection tool to screen drought tolerant germplasm. CTD was calculated by the difference between the ambient temperature and canopy temperature. Negative values of CTD denotes cooler canopy and drought tolerant genotypes, whereas the positive ones, hot canopies and drought susceptible. We measured CTD with the handheld agritherm infrared thermometer. And wilting. So leaf wilting is a symptom of low moisture stress. And we used a wilting scale of 0 to 5 to rate the peanut plants. 0 are potentially healthy plants with no wilting or leaf dropping symptom. Then 1 were some terminal and newer leaves fold up, but overall the plant looks healthy. And as we progress toward the scale, where 5 are the plants where all the leaves are severely wilted and dry, and the plants are almost physiologically dead. Then SPAD readings indicates the amount of chlorophyll present in the plant, which is related with the photosynthetic capacity and used in peanut to see the genotypic variations in the chlorophyll density among the genotypes. And SPAD data was collected at different growth stages during the season. So here are the preliminary results of 2019 field data. First, I did the PCA to reduce the large number of variables in data by extracting the important one. Though this PCA report shows that the first principal component accounts for almost 16% of variation in the data, and together both components about 25% of variation, which is not high. But I was more interested in the variables which have same magnitude and direction for different attributes like Ndvi, Ctd across both the locations. And then we performed the descriptive statistics to get a summary statistic of the measurements at different time points. And based upon the quantile, we selected the top and bottom performing lines for next year phenotypic study under control conditions. So for phenotyping in 2020 was done on selected subset of lines coming from 2019. These set of lines were grown in randomized complete block design with 2 treatment, irrigated and drought in 3 replications each. We use rainout shelters as shown in the picture to induce drought stress. In addition to Ndvi and Ctd, we use Lycor 6800 machine to collect data on carbon dioxide assimilation stomatal conductance to investigate some of the physiological adaptations that help the genotypes to perform well under drought conditions. So this timeline shows the data collection and the induction of drought condition. The trial was planted on 1st of May under 2 treatments, irrigated and drought. After 60 days of plantation, rainout shelters were pulled on the drought treatment only to induce drought, whereas irrigated regimes were kept without covering. The irrigation regimes continuously received required precipitation throughout the growing season by rain-reel irrigation system. After 45 days from the inception of drought stress, the rainout shelters were pulled off from drought regime to let the plants recover from drought. The data was continuously recorded on weekly basis for Ndvi and Ctd, and the photosynthesis data was for 2 weeks and 4 weeks after drought. So this graph shows the trend of Ndvi and Ctd in irrigated and under rainout shelter. The x-axis shows the days from planting, the y-axis shows on the left the Ndvi value, and the y-axis on the right shows the Ctd value. The yellow arrows shows the day from where drought stress is induced, and it is 60 days after plantation. The blue line represents the entries of irrigated regime and red one for rain shelters. Now we can see that in case of Ndvi, after the drought stress is applied, the entries in irrigated regime perform better than those under rain shelter. Similarly, for the Ctd, the rain shelter values are more positive, that is hotter canopies, than the irrigated one. This concludes that we have differences for Ndvi and Ctd in between the regimes, but we are more interested to see if there is a difference in between the lines or entries. So for this, we use this generalized linear model with the logistic regression in Stata software. So for any dependent variable as in shown model, this model will compare all the RIL lines with the check in both the regime for the time points of measurement. The check here we used is SPT, which is drought tolerant line, and the link and family in the model is based upon the distribution of those dependent variables. Robust and no log are the default functions in the software. So based upon the model and distribution of Ndvi, this is the coif plot for RIL lines, that RIL lines taking SPT as our base for all comparison. We use coif plot to visualize the regression model to look the pattern or trend of in our data. And the lines with the asterisk are statistically significant and good performing than SPT. Similarly, this shows for the Ctd, where few lines are significantly different on both sides of the SPT. And then this shows the same. This shows the trend in wilting. So all these lines are compared with the SPT, which is our check. So the previous graph shows the trend of all entries, and this graph will show the difference for pool by regime for wilting and Ctd. The x-axis shows high and low pool for irrigated and rain shelter. The y-axis shows the scale for wilting and Ctd. When we look for irrigated regime, we don't have significant differences in high and low pool for both wilting and Ctd. But under rain shelters, you can notice the significant differences in the performance of high pool is less wilted than and similarly for the Ctd, high pool has more cooler canopies than the low pool. For photosynthesis, we did Dunnes test to perform multiple comparison with the check. Here we have belly and SPT as our check. In this graph, the x-axis shows all the entries, including check, and y-axis shows the scale of carbon dioxide assimilation. The solid blue bar represents the irrigated regime and the red one rain shelters. The horizontal dotted lines indicate the performance of belly and SPT under drought condition. Under drought condition, when all entries are compared with SPT, most of them are significantly low, but a few lines indicated with yellow arrows perform better than SPT. Whereas under irrigated regime, when all entries are compared with belly, few entries indicate with green arrows perform better. So with all these graphs from phenotypic data, I conclude that we have few lines which perform good or comparable to our checks and can be further used in genotypic analysis for mapping. So for genotyping, we genotype the population using GBS method. We follow the standard protocol shown in the flow chart. First, DNA was extracted across all the lines in the population with modified CTAB method. Then GBS libraries were constructed by using DNA template, ligated with the optimized APKI adapter, and then barcoded. And these libraries were sent for sequencing. We used NOVA-seq sequencing platform for sequencing, and sequencing data is in process for analysis. So the future plan for completion of this project is phenotyping will be continued for validation in 2021. Sequencing data will be analyzed for variant calling using different genomic tools, and genomic regions will be identified for drought tolerance from association of phenotyping and genotyping. Finally, I would like to thank my advisor and co-advisor, Dr. Balota and Haig, for continuously helping in this project. I want to thank my committee member, Dr. Jeff Dunn and Dr. Jim Westwood. I would like to thank all the lab members helping me in field for data collection. Last but not least, I would like to thank USD and IFA who fund this project. Thank you.
Thank you very much, Naveen, for that presentation, and thanks for participating with us. Do we have questions for Naveen? Questions for Naveen as we move forward? Naveen, I think everybody's thinking about lunch right now, but an excellent presentation, and thank you for, oh, there's a question coming in now. From Dr. Soraya Bertioli. Very nice talk. The traits you are trying to map are very complex, involving multiple genes and pathways. Whereas the genomic tools will point you to genomic regions, what do you do to plan to correlate the genomic regions?
Yeah, that's an interesting question. So that's true. The focus is on drought mapping, and that there are markers that would serve as introgression or the flanking regions of interest, and we can use that for marker assisted for stronger agronomic backgrounds, because we know that in this population we don't have enough lines which are high yielding than the cultivars in the breeding program.
Okay. Thank you, Naveen. Any other questions? Okay, and your time, here's one. Were you more interested in higher physiological activity under drought or stability between well irrigated and drought within a given line? So were you more interested in higher physiological activity under drought or stability between well irrigated and drought within a given line?
So there are different approaches, and when we target for the rainout shelters or the drought, our more focus is on stability, drought stability, or the yield stability under the drought conditions. But similarly, we are also looking for physiological aspects in the lines in irrigated where they can have more yield potential as compared to yield stability.
Okay, thank you for that. All right, that's the time we have. Naveen, thank you very much.